Now, phospholipids, this is the molecule that makes up our cell membrane and is very, very, very important that these phospholipids are present because the way they're configured now, they have got two fatty acid chains, and those fatty acid chains are attached to the glycerol molecule, so there's not a third one. But there's also a phosphate group, a PO4 group, that is attached to the glycerol end. The two fatty acid tails are hydrophobic. We know that they're hydrocarbons, they're hydrophobic. But the phosphate group makes it hydrophilic. So what happens is we have, here's on the left, and one of them has got a double bond, one of the fatty acid chains. So you can see they draw it with a little kick out because it causes the kink there. And we have this PO4 group, this phosphorus group, which is here. And then we have the glycerol molecule, and then we have our fatty acid chains. Now what happens is with that phosphorus group, it makes it polar. So they call it a hydrophilic head, and these tails are still hydrophobic. They fear water, so they are nonpolar. Well, when this molecule is in water, and our cells are in water, they position themselves in this bilayer. So at the top, you have got this layer with the phosphorus group. They call it the head portion. It's hydrophilic. The tails gravitate inward with each other and bind. Those are hydrophobic. And then on the inside of our cell, we have another hydrophilic layer. So interestingly enough, if something that is polar is trying to come in, it will form hydrogen bonds with the heads. But when it gets to the middle tails, the tails are going to push it away because remember, they're water fearing, they're hydrophobic. Consequently, if something nonpolar is trying to come in, it's never going to form a hydrogen bond with the head because hydrogen bonds only form between polar molecules. Remember, water joins to another water molecule with a hydrogen bond. Well, something polar trying to come in is fine, and it's great right here because it'll form hydrogen bonds with the head, but when it gets to the center, it is pushed away. Consequently, if something is nonpolar and is trying to come in, it's never going to get further than the head because the head is polar and it will push it away. So to get something across our cell membrane is going to be a little tricky, and we'll see that there's going to have to be another means of getting things into and out of our cell. All right, this just says when phospholipids are added to water, they sort of self-assemble themselves into that double-layered structure that we call the bilayer, the phospholipid bilayer. At the surface, phospholipids, they're arranged in a bilayer as well, and the tails are pointing inward. The structure of phospholipids results in this arrangement that we find in our cell membrane. It's also the cell membrane is just like it, the nuclear membrane looks just like it. The existence of cells depends on phospholipids. We have to have these phospholipids in order for our cells to grow and divide. They have to have this phospholipid layer, and it's really, really important that we get this type of lipid in our system to form this layer. Okay, steroids. Steroids have got 4 fused rings. When you see them, they have got those aromatic rings, and there's 4 of them fused together. And they are a type of lipid, and the most common is cholesterol. Cholesterol keeps your cell membrane from freezing. It is the most abundant steroid in the human body, so you need to know that. It is also a precursor from which other steroids can be synthesized. In other words, it forms a base and then other steroids can be formed off of it. So cholesterol is really important. Now, there is 2 types of cholesterol. There's good cholesterol and bad. If you have got a cholesterol level that is over 200, your doctor will probably do a split and want to see what the bad looks like against the good. So he splits it down into what's called HDL and LDL. HDL stands for high density lipid, and LDL stands for low density lipid. High density lipid is the good stuff. So if you have this high cholesterol collective number of 200, say, that's usually when they consider it high, then they're going to split it out. If your HDL is higher than your LDL, then they generally just tell you to watch your diet, no big deal. But if your LDL is higher, that means the bad stuff is higher than the good, then they're probably going to try to give you some sort of cholesterol medicine, some sort of statin, because this is the kind that can contribute to heart disease.